This is part 2 of review part 9. We're going to talk about the specific steps of cell respiration. So we're going to start with glycolysis. Let's take a look at this picture right here. This picture shows you how glycolysis works, sort of. Okay, we start with glucose. That's what we generally start with, the six carbon molecule glucose. And then we go through glycolysis where we break the glucose into two pyruvates or two pyruvic acids. During this process, you need something called ATP to kind of activate the process as the initial spark that allows these bonds to break. So we put in 2 ATP and we get 2 ADP out of it. And then we're going to continue on and get 4 ATP out of this, right? Because we're breaking bonds, we're releasing energy. How is this energy is being released into the form of ATP. So we are gaining 4 ATP in total. And then we also make something called NADH, which is an electron carrier. How it kind of works is that we have this electron carrier that has the ability of carrying 2 high energy electrons. And then, so this electron carrier will be called NAD+, right here. When these electron carriers actually taking in the electrons, together they will form something called NADH, which has high energy, right? And then we also make pyruvic acid, obviously. So during this process, we have 4 reactants and 4 products. We have glucose, ATP, ADP, NAD+, as our reactant, and we have ADP, ATP, NADH, and pyruvate as our products. Our main product is really the pyruvate, but ATP is pretty nice as well, right? Because we can use the ATP directly in our body, and NADH can go on to like the electron transport chain and make even more ATP. So let's take a look at the questions. The question says, question number 1, what happens during the process of glycolysis? We break one glucose into 2 pyruvates. That's what happens. What are the products of glycolysis? There are 4 products. There's ADP, ATP, NADH, and pyruvic acid. If you were to just say ATP, NADH, and pyruvic acid is the product, that also works because really when you make 2 ADP, they go back into the process and get used to make 4 ATPs in total. Where do they each go after the products are made? When this ADP is made, it gets used again. When ATP is made, it gets used in the cell as a source of energy. NADH gets taken to gets transported to the electron transport chain to drop off the electrons. And pyruvic acid is going to continue to go on to the Krebs cycle, but pyruvic acid is really produced in the cytoplasm of the cell and not in the mitochondria. What is the electron carrier produced? That would be your NADH. How many ATP is invested? So that's our initial 2 ATP that we put into the system. So we're investing 2 ATP. In total, we're producing 4 ATP. It's the idea of, let's say you already have $2. You give this $2 to Ms. Bai. Ms. Bai gives you $4 back for some reason. In this process, you're gaining $4, or in this case, 4 ATP in total, but you're really gaining 2 net ATP. You're really adding on to your initial, value by 2, right? So 4 ATP is produced, but the net ATP production is 2. So if you look at the summary chart, we have glucose being used, and then we make 3 really important products, ATP, pyruvate, and 2 NADH. But really, if you think about it, if we're making NADH, obviously you need NAD+, 2 NAD+ as reactant, and if you're making 2 ATP, you need ADP as reactant as well. Now let's take a look at the Krebs cycle, the second step. Before the Krebs cycle can happen, there's an intermediate step called pyruvate processing. So how this works is that pyruvates get produced in the cytoplasm, but most of the cell respiration still happens inside the mitochondria. So we first have to transport pyruvate across the outer membrane and the inner membrane of the mitochondria, right? So if we zoom out a little bit, you can see that this entire thing, this is the mitochondria. We have to take the pyruvic acid into the mitochondrial matrix. And then what happens is that we're going to process this pyruvic acid into acetyl-CoA. So this process is called pyruvate processing. During this process, there are 3 products made. Obviously, we're trying to make acetyl-CoA, so we're going to make some acetyl-CoA. We also make CO2, and we also make NADH again. So we're making more electron carrier, taking the high energy electrons eventually to the electron transport chain. So let's take a look at the summary right here. We have pyruvate making acetyl-CoA, NADH, and 2CO2. All right. So this NADH eventually is going to go to, there we go. It's going to go to the electron transport chain at the very end. All right. So let's take a look at the questions. How many pyruvates are produced when one glucose is used? We make two pyruvates. Wait, this is not the Krebs cycle yet. How about when two glucose molecules are used? If you're using two glucose molecules, then you'll make 4 pyruvates instead of 2 pyruvates. What are the products of pyruvate processing? NADH, acetyl-CoA, and CO2. Where does pyruvate has to travel from and to before pyruvate processing can take place, right? Again, it has to travel from the cytoplasm into the mitochondrial matrix before pyruvate processing. This actual processing can take place. So I have to transport the pyruvate. How many membranes does it cross? It crosses 2 membranes, the outer membrane and the inner membrane. So the pyruvate has to go through the outer and the inner in order to enter the mitochondrial matrix. What are the products of pyruvate processing? Wait, didn't we just do this? We did this already. Oh, my bad. All right, next question. What are the products of I just said this. What is the 2-carbon molecule produced during pyruvate processing? So we have glycolysis, pyruvate processing. This 2-carbon product is called acetyl-CoA, and this carbon that we lose over here it's going to become CO2. It gets out of your body. Why does glycolysis have to occur first? Because we need to make acetyl-CoA, and acetyl-CoA, as you can see, is what's really going into the Krebs cycle as an input. Moving on, for each turn of the Krebs cycle, what is produced that can, okay, we're on to the actual Krebs cycle now. So let's take a look at the Krebs cycle right here. We have the input of acetyl-CoA, and then there are 4 things made that you have to remember. There's CO2 being made, NADH made, ATP made, and FADH2. So in this case, we're making 2 electron carriers. We make NADH and FADH2. We also make ATP that can be used directly in the cell, and then we have CO2 as well. So let's take a look at the questions. For each turn of the Krebs cycle, what is produced that can immediately, did I miss a question? No, I didn't. What can be used immediately that will be the ATP? What are the 4 main products? We just said CO2, ATP, NADH, FADH2. Where do they go after they're produced? These are electron carriers. They carry electrons. They're going to go to the electron transport chain and drop off the electrons. This is where the next process starts. You're going to take the electrons, you're going to drop it off at the electron transport chain. What happens to the carbon dioxide molecules after any of the carbon dioxide is made in your body? Your circulatory system will take the carbon dioxide, transport it to your lungs so that you can breathe it out, because we don't need carbon dioxide in our body. Lastly, can fat and proteins produce acetyl-CoA to allow the production of ATP? The answer is yes. If you watched the last video, you would know that fat and protein can be broken down and then go into the process of cell respiration as a reactant as well. Lastly, let's talk about the electron transport chain, which is where most of the ATP is made, which means it's very, very important. So let's take a look at where this whole thing takes place. If you look at this entire mitochondria, we have the outer membrane and we have the inner membrane, and you can see these large proteins located on the inner membrane of the mitochondria. This is called the electron transport chain. Okay, so the electron transport chain is located on the inner membrane of the mitochondria. We really call this last step chemiosmosis. These are the electron transport chain because they're the ones who are actually transporting the electrons. So what you see over here is that we have these electrons being dropped off by NADH and FADH2, right? We have this NADH, it has electrons, we take it out, we drop off the electrons. And then what happens next is that these electrons will travel down the electron transport chain and pump hydrogen, okay? So the idea is we take the energy of the electrons, and as these electrons are traveling from one carrier to the next, to the next, to the next, to the next, these electron energy will be released, and this releasing of energy allows the hydrogen molecules, hydrogen ions, not molecules at all, hydrogen ions to be transported from an area of low hydrogen concentration to an area of high hydrogen concentration. So we're going from low to high, which is normally an active transport process, right? We're going against the gradient, but this can still happen because the electron energy is allowing this to happen, right? At the very end, so you see these electrons moving, this is the yellow line. At the end of the electron transport chain, the electrons get combined with oxygen. And this is really important because oxygen combining with the electrons is what's taking the electrons away from the system, right? So think about you drinking from a straw, right? The water won't come up the straw unless you're actively pulling the water away from your cup, right? So these electrons can only travel through the electron transport chain as they combine with the oxygen, and forming water. The electrons cannot go through the electron transport chain if there's no oxygen. So that's the purpose of the oxygen, is the final electron acceptor. So I'm going to write down the process of this energy transfer. We have NADH and FADH2 that has energy, right? It's going to transfer this electron. This electron energy gets dropped off, right? They get dropped off into the electron transport chain at the ETC. All right. So as these electrons are losing energy, the electron energy becomes, so this is the energy transfer process, what I'm trying to say. As these electrons are losing energy, the energy gets used to pump hydrogen and is low to high, right? So we're going from low to high, low concentration to high concentration, from the mitochondrial matrix to the intermembrane space. And then this pumping of hydrogen, eventually, what is the point of spending all this effort pumping this hydrogen, moving electrons around? The point is, these hydrogen can go through the ATP synthase, and because the hydrogen ions are going from an area of high concentration to low concentration, this serves as an energy source as well, because think about water going down the hill, right? If you put a turbine over there, a little electricity generating machine, blanking, whatever that word is called, right? Then you can generate electricity. You can turn the water power into electricity, right? Same idea over here. We have a lot of hydrogen over here, not so many hydrogen over here. And as these hydrogens all flow through, this ATP synthase can spin and put ADP and phosphate group together to make ATP. So we pump hydrogen low to high, and then this hydrogen concentration gradient allows ATP synthase to spin, right? So we're transferring the energy of the hydrogen into another form of energy, the spinning of the ATP synthase, which allows ADP and phosphate group to combine to make ATP. Okay, so as you can see, this entire process, every single step contains some sort of energy. We take one form of energy, turn it into something else, power something else, and then eventually we make a ton of ATP. All right, so take a look at this one more time. If there's any part of this that you don't understand, please pause and go back to the earlier part and listen to what I was saying one more time. All right, so let's take a look at the questions now. Questions are right here. Oh man, it's been a hard day for my computer. All right. Okay, so let's take a look at the questions. We are looking at the electron transport chain. What event takes place in the electron transport chain? Electrons get dropped off by the electron carriers. Eventually we make ATP. Or you can talk about the entire process, but really that's what I'm looking for. Where does the electron transport chain take place? You will say the inner membrane of the mitochondria. Obviously, all the parts of the mitochondria is involved for cell respiration, but the electron carriers in the electron transport chain is located on the inner membrane of the mitochondria. That's exactly what it says, right? Where are the electron carriers located? What molecules drop off electrons at the beginning? Those are our electron carriers, and there are two types of them. One is NADH, one is called FADH2, and they come from previous steps: glycolysis, pyruvate processing, and the Krebs cycle. The next question is, when electrons travel through the electron transport chain, what molecule go through the electron carriers and getting pumped from low to high That is the hydrogen, right? Going from low to high. Electrons pump. Just do this a few times. Maybe you'll remember it for the test. What molecules are traveling through the carriers, right, and getting pumped. So when these electrons are traveling down, we pump the hydrogen from the matrix to the intermembrane space. So when this molecule is pumped, this hydrogen is pumped, during the electron transport chain, which area has a low concentration of molecule? That will be the matrix. High concentration of the molecule will be the intermembrane space. And this gives you a charge difference as well. When you have less hydrogen ions that carry positive charges, that hydrogen ions carry positive charges. So if you have less of it in comparison to the other side, you would have a partial negative charge in the mitochondrial matrix, and what did I just say? Partial negative? Sorry, negative charge. In comparison, you will have a negative charge in the mitochondrial matrix and a positive charge in the intermembrane space because you have a lot of these positively charged hydrogen ions. The purpose of the oxygen is to serve as a final electron acceptor. It takes the electrons, it slams it into some oxygen, take it away from the system so that we can keep on pulling the electrons through the system. All right? Why is it necessary for you to breathe? Because you need oxygen for cell respiration, and you need to get rid of the carbon dioxide because you don't want carbon dioxide in your body. It's useless. What is the final electron acceptor of the cell respiration? As we just said, that will be oxygen. Why is the final electron acceptor significant? Because it pulls electrons through the electron transport chain, right? If you were to look at what I wrote on the bottom, if we don't have oxygen, then the electrons cannot travel through, which means the hydrogen cannot be pumped, which means hydrogen gradient doesn't happen, which means ATP synthase doesn't spin, which means there's no ATP made, right? This is awful. If you're not making ATP, you're not going to last very long, okay? Not okay. You have to remember this. Oxygen keeps you alive because it pulls the electrons over through the electron transport chain. One molecule is formed after oxygen combines with the electrons. That will be water. You have some oxygen, you have some electrons, add a few more hydrogen ions, you make water, right? Producing water is not the purpose of the electron transport chain or the cell respiration. Not really, right? ATP is important. Water is good to have. We're going to make it anyway, okay? What molecule is formed? That's what we just said. What causes the production of ATP from ADP? That will be hydrogen flowing through the ATP synthase, causing ATP synthase to spin, which makes ATP. One glucose generates around how many ATP in total? Around 34. The molecule in charge of powering the ATP synthase in the electron transport chain is the hydrogen ions. Again, hydrogen ions are the ones that travel through the ATP synthase, not the electrons. The hydrogen ions travel through the ATP synthase from an area of high concentration of hydrogen to an area of low concentration of hydrogen, from positive to negative, and making the ATP synthase spin, which makes ATP. This process right here is also called chemiosmosis because it's also almost like osmosis, right? Osmosis is high to low of water. This is high to low of hydrogen ions. What area in the mitochondria is this molecule traveling to? That will be the matrix. Dun dun.